Okay, guys, we're gonna get started with the CRISPR kit.
Okay, is the camera here? All right, hi everyone. We are CRISPR Kit, and today we're really excited to share with you what we've kind of been working on and what we kind of see in the future. Our mission here is to democratize biotechnology through a scalable education. So we're going to kind of talk to you about how a CRISPR kit is conceived, how it works, and then you'll have an opportunity for those in person to try out the kits for yourself. So before we get started, I'm going to do some introduction. So my name is Andre. I am a second year PhD student in Howard Tiam's lab, and I am in the bioengineering department.
I'm Maren. I just recently started as project manager for this kit to get this more in the world.
My name's Alex, and I'm a postdoc in Stanley Chi's lab, and my background's sort of like CRISPR and microscopy. So, yeah. And we just have to talk about funding really briefly. CRISPR kit's really been a collaboration with both corporations like Amgen and also a variety of organizations at Stanford, such as the Accelerator for Learning, Makers, and then also NSF. So, cool.
Yeah, and so kind of zooming out into kind of where we are today. We are in the age of biotechnology, so we have a lot of modern biotech innovations such as AI in therapeutics and gene editing, and one of the arguably the most major breakthroughs in the last decade is this technology of CRISPR. Can you click the screen? Okay, cool. CRISPR, which is a really powerful gene editing tool. And it really has revolutionized a lot of spaces in research and in healthcare. For example, Casgevy, if you've heard of it before, is one of the first CRISPR-based sickle cell anemia therapeutics, and it's been working pretty well. And so, just as a refresher for those of you who aren't very familiar with CRISPR, the way that CRISPR edits genes is that you have two components. You have a Cas9 or a dCas9 protein, and this will cut the DNA. But you need a guide RNA to also guide the Cas9 protein to its directed spot so that you can cut the DNA and edit it. One thing that's very important is that for our CRISPR kit, we actually use a dCas9 protein. And D here stands for dead. And so basically, instead of cutting the DNA, it just sits on the DNA and prevents transcription from happening. It physically blocks the polymerase from transcribing your gene. And so this technology we call CRISPR-I. So the I stands for interference. And this is also what CRISPR kit is based off of. Okay, so going back to this idea that biotech is changing. It's a constantly developing and evolving field. We see that there's a lot of advances being made, but there's a mismatch in education for biotech. So usually when you're in a biotechnology classroom or biology classroom, a lot of this knowledge is very theoretical, and there's not a lot of opportunities to do a lot of hands-on biology. And so for students, that can feel a little bit kind of far away from them and kind of hard to engage them. And so, yeah, we kind of thought oh, maybe, we can, what if there's a way that we can bring hands-on biotechnology or bio education experiments into the classroom and really help inspire the next generation of scientists. And so that's kind of what led us to develop our mission. So making biotechnology accessible to everyone for building the next generation of young scientists. Okay, so now we're going a little bit into how CRISPR kit works. So on the left here, the first three components is kind of your kind of classic CRISPR-I system, and then one additional component here is the cell-free system. And so usually when we do CRISPR experiments, we do these in live organisms. And that kind of poses a challenge because one, you have these biosafety issues, and two, you have to spend a lot of money to keep these cells alive. And so the cell-free system is actually a very important and key part of making CRISPR kit very frugal and very safe. And so yeah, a cell-free system basically is just you take take a bacteria and has all the machinery you need to translate the gene and make your protein, and you basically just extract that from the bacteria and put that into a test tube. And so yeah, it's a very cool technology. And so kind of like evaluating how like we want to make CRISPR, which usually costs like thousands, hundreds of maybe 10,000s, hundreds of thousands of dollars to something that's very cheap. You also have to think about how we can take this lab experiment and maybe scale it to something that is more accessible and more cheap to use. So for example, like with pipettes, we can maybe replace those with inoculation loops. Or, instead of performing the experiment at 37°C and requiring an incubator, you can do this at room temp. Or, instead of having a plate reader, we can use a smartphone app, which we'll talk a little bit more about. And then also, instead of using professional software, can we use like kind of open source technology? Okay. So I'm going to hand it off to Alex, who will talk a little bit more about kind of our history and kind of like the iterations we're kind of working on.
So I guess one, I think, amazing thing about CRISPR kit is that a typical CRISPR experiment might cost on the order of hundreds of dollars to do. And so if you're thinking about bringing a technology into a classroom, it becomes kind of inaccessible in a sense because, teachers, like high school teachers, can't afford $100 experiments in order to teach a CRISPR technology. So as Andre mentioned, we developed sort of infrastructures that allowed us to bring down hundreds of dollars to only $2 per CRISPR kit. So this is a price point which a lot of high school teachers can afford, but also that we can self-fund in a sense. So we'll discuss a little bit like what CRISPR kit is. So CRISPR kit's a sort of simple idea where Andre mentioned we're using this cell-free system. This is really important for us to use because it can produce proteins like a living organism, but it doesn't have the like safety or even ethical issues that using other things might provide with a CRISPR system. And one important aspect is we always use some sort of visual sort of readout in order to very explicitly show that a protein, for example, is being produced or have some sort of like molecular like sensor so that there's an easy way to sort of actually see CRISPR in action. So we work with what's called plasmids, which are DNA, and we're encoding proteins that are essentially different colors in these kits to be produced by the cell-free system. And as mentioned, this is a CRISPR technology. So we have the dCAS protein, which is then guided to the right location in by the guide RNA. So in our experiment, when we do our CRISPR experiment, if we just produce the protein, it'll produce the colors. But if it's the CRISPR system, the CRISPR turns off the color. So we can see sort of like the molecular details of CRISPR in action. And so our first generation kits were very straightforward. They were essentially what we call color kits, where we're able to produce in the cell-free system red, yellow, and blue proteins, as shown in these solutions. And then when we actually apply the CRISPR system, we're able to sort of deactivate the color from being produced. So this is sort of a visual way of seeing the CRISPR in action. And we can also sort of extend this a little bit further, such as doing a multicolor kit or a dual color kit, where we can essentially mix the two color plasmids, such as the red and yellow plasmid, to produce an orange readout. And then when we apply a guide RNA for either the red protein or the yellow protein, we can sort of deactivate orange to red or deactivate orange to yellow. And so this work was published back in 2024 in Nature Communications. But I think we also developed a few other iterations of the kit beyond just sort of this red, yellow, and blue kit. We've also produced a melanin kit, which produces melanin as a color readout, or a GFP kit, which is a fluorescent protein. And so one question is like, why would we want to create sort of like other colors of kits? And so I think one consideration when thinking about implementing educational technology into classrooms is actually making sure that your technology fits with existing curricula that you're actually trying to implement. And so teachers might be wondering, like, how do we, implement CRISPR kit into our classrooms? Well, one way that we've actually been going about doing this is by using sort of relevant AP bio curricula, such as, metabolism pathways, such as the production of melanin, and by essentially developing a kit where we can sort of block the production of melanin in this metabolic pathway using our CRISPR-I technology. We're able to sort of provide teachers ways where they can try new technologies and experiments and, without significantly modifying their sort of classroom structures. And today we're actually going to be sort of showcasing our GFP kit today, which is essentially the same thing as the color protein kits, except instead of creating red, yellow, blue, we're going to be producing a green fluorescent protein. And essentially, CRISPR kit is a very simple experiment where we're going to give you 4 tubes. One is a negative control system, where the negative control represents a system that essentially produces no fluorescent protein. So all we're going to be giving you is the cell-free system. We're also going to be providing a positive control system where the GFP is produced without any CRISPR in the system. And then we will also be providing both a system that contains a guide RNA and Cas9, essentially the CRISPR machinery to turn off the GFP production. And then we also have a no-guide RNA condition. And the point of the no-guided RNA condition is to sort of go at this idea of like off-target effects of Cas9. Does this protein still affect the production of GFP even without the presence of the guide RNA? So we have 4 sample conditions where we can sort of understand various questions in CRISPR using the kit. And as I mentioned, we did publish this back in 2024, and one of the main questions is like what do students think of the CRISPR technology, right? So really 2024 and 2025 has been almost completely dedicated to bringing our technology into classrooms and getting feedback and seeing its impact so that we can sort of generate next generations of CRISPR kits that are actually beneficial to students. And so we've already piloted this to over 1,000 students back in 2025. We first started locally at like Menlo School and Los Altos High School, but now we're currently we've expanded domestically to as far as like Boston, for example, and partnering with Boston University. And we're also have plans for international piloting this year, which has additional logistical complexities. And we've also pitched this to Khan Lab schools and Khan Academy, where we actually taught this right in front of South Con. So this was a really sort of exciting moment where we were thinking about bridging, hands-on frugal technology with platforms for education that are also based off of, free sort of, accessible ideas. So, and we were also highlighted at the School of Engineering Centennial event where we, I believe, gave out over like 1,000 kits to students that ranged from like 5 years old all the way to, grandparents. So this was a really exciting showcase of our CRISPR kit technology. And so our goal this year is to reach 10,000 students, and 10,000 students is a number that I think isn't really practical for a lot of academic labs to actually do, where we can't physically go to classrooms and teach 10,000 students because, this is just a volunteer activity for me at the end of the day. This is not my main role. So like developing structures where we can actually get our technology into classrooms and have teachers autonomously teach CRISPR kit is sort of what the 10,000 represents. And this requires us to, in our particular case, partnering with various industry partners to help us produce CRISPR kits, establish teaching networks so that we can actually get these into a variety of classrooms, and then funding and distribution networks. And so when I was thinking about CRISPR kit's a way that we were able to bring CRISPR down to $2, but there are a variety of different challenges when we're actually sort of thinking about how to make a technology actually go into classrooms and be effective. So I have this model that I call sort of like the accessibility in 5 dimensions, where essentially there are 5 dimensions where you can innovate in sort of this educational technology space. in order to sort of improve the technology beyond just the science. And so like CRISPR-Kit was originally founded on this idea of the financial model where, CRISPR's a few $100, we're bringing it down to $2, but other things that, might be barriers for access in the classrooms could range from logistical issues, such as if it requires freezer space, then suddenly, it prevents certain teachers from being able to use it. Pedagogical in the sense of like, we have to make sure that our, kit actually fits various curriculas and teachers find it useful. Cultural, some people might find the color kit just isn't interesting enough or motivational enough in order for, to be interested in using it. So developing sort of ideas that sort of fit various, other ideas are ways that we can sort of expand our CRISPR technology. So just briefly, we've developed various strategies, such as we use lyophilization to make our kits totally shelf stable. And this allows access actually to sort of international countries. We no longer have to ship it on ice, which brings down the cost, but also, keeps things lasting long term. We're currently developing an AI app system where students can actually analyze their CRISPR-Kit experiments and actually get in-classroom assistance using our AI chatbot. So if anyone's interested in trying that out, we're currently piloting a study for doing that, and then also sort of developing curriculum-friendly designs and teaching organization partnerships. Nice.
I was just seeing if there's any questions.
Okay. So yeah, as I mentioned, we're always thinking about sort of the next steps with CRISPR-Kit and what a next generation CRISPR-Kit might look like. So, CRISPR-Kit's just a kit that sort of changes colors and we can sort of see CRISPR in action. But I think students are motivated by the idea of like agency and being able to sort of like being able to affect and sort of change, I guess, research and science and add to that. So that's sort of our goals for these sort of like next generation CRISPR kits, where we're trying to connect relevant and diverse topics to classrooms. And, when we think about CRISPR, such as what Andre, we often think about it as a medical sort of technology, but CRISPR has technical impact much beyond that, such as in agriculture, sustainability, even basic sciences like chemistry, and sort of developing kits that can sort of go after those different interests might be a impactful way to sort of get it into classrooms. And all we're doing is connecting a simple readout, such as a color or a mass change or something like that, with a genetic concept of interest, and linking those two together to create sort of a next generation kit. And so, as discussed, like it would be great if we could even extend this for high school students to actually use CRISPR-Kit technologies to make their own sort of scientific discoveries in the future, and maybe have sort of a customizable component in order to go after those questions. And so I think CRISPR-Kit is more than CRISPR education. So I was sort of like inspired actually by sort of Foldscope, and I think that, when we think about the 20th century sort of educational technology for STEM in particular, like a lot of people, like Nobel laureates, reference the chemistry set or the model telescope or microscope for their inspiration for wanting to do sort of science as a career. And so I think sort of frugal science right now is really important, because we need to sort of connect like impactful technologies that we're producing at Stanford or at other universities and bring it into classrooms so students know what scientists are doing right now, so that they can be motivated to want to become scientists also. And so what we're really doing is using this idea called like educational transfer, where scientists sort of translate their technologies into sort of accessible formats so that students can actually learn these technologies like CRISPR, or like in Foldscope's example, like microscope design. So really sort of our goal is to make frontier biotech physically accessible, not just conceptually accessible. And so, like, I just posed this question, like, could frugal platforms like Foldscope and CRISPR-Kit define the 21st century of, STEM ed tech, like sort of the chemistry set did in the 20th century. And so we have 2 students in the class today. So each one of you is going to get to be able to try your own CRISPR kit out person. Yes.
What innovation actually allows you to do CRISPR-I experiment 100 times?
Yeah, that's a great question. So I think one of the main challenges is actual the cell culturing, and the equipment that's involved in actually doing these experiments are pretty expensive. And so our cell-free system is actually our most costly component, but it's also what's bringing down the price of CRISPR kit most significantly. So
you're in that calculation, you're saying in order to do regular CRISPR-I, you need a full lab. You're saying
yes.
Is the kit basically you grow bacteria that are expressing Cas9 and the thrust proteins, then you just splice them and or is there
Oh, it's actually even simpler than that. So there is no growing. We already have the cell-free system is already totally functional. So all you have to do is stick a gene of interest in.
But I mean, so how do you generate that cell-free system?
Yeah.
Is it basically bacteria that are expressing everything you need in a cell-free system, and then you kill the cells?
Yes, that's essentially what it is. We just remove the membranes and just have everything else left over. So, and this allows for large scale production also of the XREX system. So, does anyone have any questions before we try out the experiment? So, yes.
Beyond education, do you see this having other impacts, low income settings?
So I think that CRISPR kit is uniquely well positioned for low impact. I meant Title I schools in particular, because I think a lot of school districts that are underfunded also don't have the ability to run the same level of experiments that the high schools that we've piloted at have. A lot of these high schools have pipettes, a lot of equipment to run I would say more modern science experiments, but CRISPR kits position where you actually don't need any of that infrastructure, such as pipettes, to even run the CRISPR kit. And we're basically giving out CRISPR kit for free. So we're actually most interested in bridging Title I schools and low income school districts and teaching them about these kinds of technologies that exist. Yeah. Did that answer the question?
Sort of.
Yeah, sort of.
I guess as a follow-up, for example, with the fold scope,
Yeah.
it's like a microscope for low income, or it's a very cheap microscope, but it's also being used for malarial diagnostics a little bit. Or it's being tried out for that. Is there some kind of equivalent with the CRISPR kit?
No, but it's a great follow-up. It's we're we're much earlier stage than Foldscope, which has been around for, I don't know, 15 years now. So we're at year 2. So it would be great if we could use a color-based readout as a diagnostic for maybe low income settings, right? But we're not at that stage yet. So if you have ideas of how you could actually translate that into a CRISPR kit, we would definitely be interested. So
I would also say the biggest the dream is to take any gene you want and do a CRISPR-I experiment on it. Right now, we just have colors and melanin and GFP, but if you can edit a gene of any interest, that can be very cool. And if you can do this at low cost, right, then you kind of are advancing the field of citizen science, right, where anyone can do an experiment and advance our scientific knowledge. But that's the dream.
Thank you.
okay. So if we have no further questions, let's get started. So the next generation of CRISPR kit, this is the experiment we're going to be doing. So how this is going to work logistically is we'll just come up here. There are only 2 of you. So
TAs can also do it.
TAs can do it
Okay,
also.
cool.
And yeah, we'll hand out the protocols. So
Here we go once again. Okay.
How do you want them to do it?
No, we'll just do it side by side.
Well, how do you want in a second? I only.
Okay.
That's not a good thing.
Oh, make sure before you start, please wear gloves. Grab gloves.
Where do you want to go anymore? Just one.
Oh. Yeah. Need a update the logo.
What is the
How long can
lowest
you, once you've
age
prepped your samples or your reagents, how long
deducted
does it
this?
survive? What's the,
It's probably the last one.
We've had like children to children. do it. Yeah.
Probably like where is that? The viewers.
I think they do. I mean, in terms of doing an experiment, it's the youngest, but we've had like sixth graders.
Because the software system
And I
is
think
no ribosomes.
if you can explain CRISPR in a very simple way, it's like, oh, it's a way to target DNA,
Yeah.
then
So we haven't like studied yet.
bring it to a lower sort
Like
of
all the
bracket.
processes, so I see what you envision is.
I see.
Like I'm saying bracket, that this is like, not just like, oh, colors.
Middle school is when I learned biology, so maybe around that sort of like age range. Do you think it could go lower?
Oh, are you still there?
I mean, I guess it depends on what associated.
so everything here you synthesize making lab?
No, we actually.
Okay, yeah, I've been trying to pilot it actually in elementary schools, but
Really?
then we have to sort of work with elementary school teachers to adapt everything, so.
Yeah, I mean, I feel like old school, like the conceptualizing a microscope is, in my opinion, much easier than conceptualizing CRISPR.
Yeah, I actually ran Foldscope for 5 years back when I was a grad student at Berkeley.
Mm.
And I feel like CRISPR kit's a little bit too complicated for the like K through 5 probably,
Right.
but
Maybe we can with the camera. So.
Oh, yes.
Oh, yeah.
Do you want me to-
Yeah.
Okay.
Experiment.
We're gonna do a live demo.
Oh, does it actually have? Oh.
Yeah, and nobody else stayed fast enough on it, no?
Are there any questions on Zoom?
Oh.
There you go. Okay.
Oh, sorry.
No, I just got a quick question. So the negative control
Yeah,
will remain the same color as it is now?
it'll be clear,
Clear.
right? Because you don't have anything in there.
Okay. If positive control has,
We'll give you color.
okay, and that's, we don't need to add anything to positive control.
No,
Okay.
right?
So what you have to do is like the Sapri system and the water is already in there. The first thing you have to do is like add the GFP plasmid,
Oh.
and I also label like you can see in where you have to add it. You
Mm,
basically don't add it to the negative
Okay.
control, but then the positive, and the no guide and the guide control, and then you add your CRISPR I components, so the DCAS and the guide RNA. DCAS you add in. If you go here, you add in like the no guide and the guide.
I
And
see.
the guide RNA only in the guide.
So the guide will have no color in the end, and that's a positive reaction.
That's actually what we want to see. So
Oh.
this is basically these
Yeah.
are all controls,
Yeah,
and this is the sample. Like this is the condition we're actually testing for.
Okay.
So if it works, yes, it is not colored. If
Okay.
not, that's something okay.
okay. And how does the GFP positive control work?
So
Like, I don't understand how it
because
becomes
it's a
green.
plasmid and like in the cell-free system, like it's gonna express.
Oh, because there's the
And then-
ribosomes.
Exactly. So then that's why you get the color. In the no guide, you also have the same thing. If the decastinate doesn't do anything with the plasmid, it might have not like off-target effects, but so far we haven't seen that. So this one should have basically the same control, like should be have like around the same color as phosphor control
I see.
because the DCAS9 shouldn't do anything by itself.
Right.
Only in the combination it should.
But I don't understand how, like, the color green, right? Because this is a fluorescent protein, right? So I'm like excited with blue light fluorescence. Like, is it by
It produces so much protein actually that you can see it absorbing and fluorescing actually.
Oh wow.
So it turns green.
Yeah, it's actually like really.
Under the like a UV light that you should see person, I think.
Yeah.
So do you need sunlight?
No, you don't even need it. It'll basically autofluoresce.
So it will like take a couple like hours.
Does it need, like,
But if you put it under the UV light, so basically
Oh, nice.
these are like the negative control, the positive control, the no guide, and then the guide.
yeah, but it's still like, you have to like check concentration.
But this is like if you put it under UV
Yeah.
light,
And like, I mean, ideally,
that's
yeah.
basically your full.
And
And then you see like it's a tiny bit, like a tiny bit. That's like, but like the guide really like lowered it. So like if you compare to normal control, you see like.
Oh, I see. Yeah. Cool.
So, but like
Imagine you diluted it.
if you look,
Three times, and then you added like
this is how.
ag or some agent. So you have to increase, so that it's like. Effectively like non-standard.
And
Yeah.
another question on the cell-free system. So there's ribosomes and like ATP. And what else is in the?
What exactly is in the software system?
It depends what you're like.
The transcription.
It's basically everything without DNA
Lower resource.
in RNA, and we sort of precipitate that out. So it has like ribosomes, a lot of proteins in it. And yeah, you can do more than transcription and stuff like
Okay.
that. So
Right, but what I'm wondering is the ways
it's a
to.
cool system. So Michael Jewett in BioE is actually who we collaborate with. And so they do a lot of different cool sort of like experiments there. This is just like a simple adaptation.
I see.
Literally that it is.
Okay.
Even if that's good concentration.
Yeah, cool. You want to do that first?
You guys go. You guys go.
Sure.
Yeah, go ahead.
Yeah,
This is my favorite. It's that's one of my favorite.
yeah. It's, yeah.
If one of you can create more GFP, then your pipetting skills maybe are better than all the other two.
It's the competition now.
Yeah.
Yeah. Well, those be worse because we're pipetting too much.
Maybe. Yeah, who can create the best CRISPR kit, right?
Oh, wait, that's the.
Which one is this one?
GFP goes into something.
Let's see. So we're looking, this is a GFP, like it goes in 3 tubes.
Just an interesting. Okay.
Sorry, I don't have anyone's faces in there.
That's fine. You can.
Yeah.
You take pictures. We recorded you guys.
Yeah.
Don't forget like to switch the packets.
Yeah.
Otherwise you're
To-
transferring the garbage.
Oh, it should be in the.
What is your app of like? Basically.
Yeah.
Oh, if you're interested, we need people to actually try it out for us. You want to?
What?
Could you email me and I'll send you the full instructions. We could also send it to your class, actually.
Yes.
Take the.
I can like post an announcement.
Yeah. I can,
The signal's pretty.
Andre, can you swap the email list?
Yeah.
If you want to write a message, I'll just like post it.
Like, that'd be great.
Yeah, for sure.
To just support you in doing the experiment, and it's also like manage to analyze, like optimize for like denial to the.
Oh yeah, that was. It went up to the first.
Okay.
Switch stop packets.
Oh, yes.
But should this be like pointing
Because
upwards?
basically, otherwise, I mean, like the good thing is you don't have to go this way, so like you don't want to transfer.
Sorry.
You don't want to transfer anything from this.
Oh, yes.
Like this. I think it's probably still bulletproof because you're like, this is your experimental control, unless you go this way, this way, this way.
Mhm.
I guess we can.
I mean, there's other ones that you don't know.
Yeah.
Oh yeah. and there are other ones.
A
P2
P2.
and a P.
No, it's just a P2.
Yeah.
The only pipetting 1 microliter.
They haven't sent me the.
You can also just use mine. Oh, you finished?
Oh, we're gonna.
Yeah, I did these three. Oh, okay. Okay, but you
No,
can do
I'm
the.
gonna be fine. I'm just nervous because
Okay, sick.
I have fun.
Yeah, yeah. So.
I'm not nervous. I mean,
We're just gonna do it or not? I'll just. Yeah, yeah. Just take the. Yeah, yeah.
That's good.
like I just like it here. It's like what if we forget
That's fine.
that?
Or just. I think we're good now. Yes. Take it. I don't know how to play that. I'll be fine. Well, aren't you a PhD?
I think it's better
I want
than
to
not.
though. Wait, can I use that one actually? Just one.
I agree, but at the same time, I feel like it would be.
Well, I think we're using the wrong
If it was slowed a little.
Yeah, it wasn't
I
the. Okay, let's stick to it. Okay.
That's fine.
I don't know what I'm doing. Okay, this is a PhD lab.
Actually, I don't. I think my parents say I speak like Taiwan.
This is.
So they're like.
Did you
"We can't
label all these
understand
yourself?
No, they were
Nice.
like, if you go to Beijing and
So that's
like
not
that.
like that's
you're
for
gonna
now the planet.
get-
It's so like skipping like more.
Like,
Whoa.
Wait, so I go once
You'll be
or like
Yeah,
Like,
you
once you like pick it up,
Yeah.
and you like
Yeah.
pick it up, put it in your
Yeah.
shoe. These are yours, I guess.
we'll do it together.
Oh, yes.
Oh, okay, same.
Yeah, because
sorry.
it doesn't like it lasts only for one.
Oh, okay, okay. Okay, Ryan, you do it. You can finish yours.
No, it's.
We have one more question.
And now it's actually competition.
We've had 5-year-olds successfully do this, so no pressure.
Really?
Yeah.
Wow.
Wait,
Yes.
we can be quantitative on who has like the best guy.
Whatever competition you guys want to do, I'll even
Yeah,
give you
who has the best pipetting?
I'll even give you the app. You can read out like
Okay, that's what I'm saying.
That's the thing is like,
To like switch
it's that,
tips.
actually, yeah, Alex made a good point. Alex made a good point about, pipetting small volumes is
One by
pretty
two.
difficult, especially for like high school students. Like that's.
Huge.
You have like each
guy.
I mean, it's something that we're still like,
we sort of make it
but
so that
cats you put into
even if this snaps up.
Okay.
like when
Yeah,
I
it should still give you-
yeah,
It's more.
Okay, that's what. Oh wow, this is kind of difficult.
Right, how often do you price that?
I know that every day.
The
Really?
deposit the volume into the liquid.
Are you just trying to suck this out?
You need to spin things out.
I woke up this morning.
I think like.
Oh yeah, yeah. I expect this to happen.
I tested it yesterday and it was so.
Okay, so now I have some bubbles. That's not good, probably.
That's okay.
You'll still see. Is this your positive control? Negative? Which
It's positive control.
you'll still see green. It's fine.
Do I need to flick it or no?
Just in the end, there's like a step
Oh, okay.
where you put you mix the tubes. You can pipe it in or flip it, whatever. That's really.
Okay.
It works with flipping. Yeah, I feel like these are the wrong tips on.
Yeah, because Enrico might use it by accident.
And with the inoculation loops, the same thing.
I think that they did work, but they're like a little bit less precise. So if you have pilots, I think it's a bit easier. To,
Yeah.
like, it's like the competition
What's that?
starts between one like what she gets a grade. No, which grade is
Cool. Yeah, he was my academic advisor. I'm a freshman.
Yeah, you told me that.
I did it. I don't know. I know.
This is so wasteful. I never bought them. That's insane.
Oh, that's on.
Oh, I just spoke with Brent.
I think he wrote a little bit.
It's all for you.
Maybe. Not for grad school. I feel like for some for other things.
Is everything clear?
Yeah.
Yeah.
So you added everything now?
But what?
Hopefully not positive control.
Huh?
Hopefully not positive control.
Like, points.
It is.
Have you guys seen her? I don't have.
It should be positive control.
I tried it like 2 times a lot to read them.
Oh, that should be green.
She just got hired at Oregon. So I was like,
Yeah. So.
What's that technique?
Try not to make any bubbles.
no, you can't call him secrets, Andre. They're competing.
Yeah.
Which one you are?
Oh, two.
Maybe
Really good at having sense skills.
like because like you know like you can calculate how much is in each view.
Interview.
They're not
And
gonna
then
stay
like
like.
if it's like it's basically like your one should be 8 microliters, so you can go with like a 6 microliter up and down. Then you fill the whole straight in the bottle.
Oh, like.
3 to 24 hours.
Yeah,
They're-
I mean like it depends a little bit like how much we add up the plasma. If you can get the more microliter, I can turn it in like 3 hours.
So how do you run these in classrooms? If it takes 3 hours to.
Usually it's a 2 day.
Oh, I see, I see.
Then you
But
add,
that is one thing we're trying to do too, is like, can you get it made out in 1 hour?
Yeah.
That'd be really cool.
Okay.
And it'd be nice for like a workshop.
I do.
Andre, can you also use the small tips for the P20? Yes, right? The P20 to actually the same one, right?
Yes.
Like my second time football.
Like ever?
Yeah.
They don't make you ever do this?
What about other?
Wait, what?
Yeah.
Yeah.
No, but I'm in the physics department.
Oh,
Yeah.
it's a bit nonsense.
What's your project on by?
Yeah.
Is it secret?
Microscopy.
Microscopy?
Yeah.
Oh, are you
Yeah.
building a microscope?
Yeah. Right now, like a ocean microscope,
Oh,
underwater microscope.
have you gone on an expedition?
I have, yeah.
Use your microscope?
Yes, but I got there and it broke. I made
Oh,
one,
You
so
mix it
I'll
up.
try again. Oh, I forgot. What else tried to do?
Yeah, try to like now you have like a lot of.
No, I actually forgot. Yeah.
I try to like don't drink too late. I think it should be still fine, but
Okay. How do I avoid friends?
so basically you have 6 microliter like in there.
Yeah.
Yeah, so you go in. You take like you go all the way in, all the way like to the bottom.
Wait, like all the way to what it's
Oh,
on?
sorry. You go one,
Yeah.
press one, and then you go all the way to the tube.
Yep.
You take out like as much as you can,
Still don't have
and then you just slowly go down again,
Slowly.
and then up again, and down again, and up again, and then that's good enough.
You know what? It's on the.
Yeah.
It's organized.
So what's happening?
It's so funny because okay, this is like
Yes, and now it's
through
shooting
Harvard.
equals.
Oh, you're playing all that time.
student organization.
Are you gonna hit the interviews with?
But I use pipetting in the sports. I usually use no.
Or in this course?
Yeah.
Yeah, I also.
Okay.
How do I fix the balls?
So basically, you go like you press some.
Up and down.
Yeah. Well, wait. So you press already when you go in. You go all the way to the.
So are you two undergrads?
Mhm.
Have you taken 44?
I haven't.
Like which?
But my friend who's online said that you were.
Yeah, that's not really like helpful to.
Oh, really?
Yeah.
I should make it more.
I'm not sure.
Nikita?
Oh, yeah.
Let go.
I know her.
It's like all the.
What's
Let go
44?
out. Like never like.
It's fundamentals of engineering biology. So you're in this lab, you're like here and you basically do, like you learn how to do a lot of like bioengineering techniques like
Me too. So
cloning, transfection.
I need to-
like a volume of 8 microliters, and then you
Like very
Electrical.
60 you can.
Are you bio it?
I'm fancy.
Then we like take it down to get the liquid down.
Oh.
Because sometimes you have like liquid on the top and it's just have like your hands and use.
I just felt the centrifuge. I don't even to.
Oh, really?
Mhm.
Oh, there you go.
Right over there. Get it.
Wait, who's taking that class? Building the centrifuge?
Mm.
Are you taking it?
Yeah, I'm taking it.
Who's TAing it?
Oh, a lot.
Oh, a lot.
Let's see.
Okay.
I'm gonna feel bad for missing.
Oh no, it's okay. I just want to see if I know anyone.
Did you
Yeah.
order anything?
No, I'm
Ginger.
missing the water.
The water's already, seasons in the
oh.
water's already. prepared.
Antoine.
We have to do this.
KB.
Did you do all three?
Dog.
Yes.
These three? Okay.
But I
Kevin.
Kevin. Some bubbles.
That's okay. Bubble like that can sometimes come with it.
No.
Okay.
Like,
I know,
way more.
I know Anton, KB, and Kevin.
Yeah.
They're all in my cohort.
Made like lower stuff.
I heard it's a lot.
So in the reaction has like that.
It's a lot. I've spent so many hours
You
on
have-
this week.
I think you have like a total of 70 assignments this quarter.
Are you serious? I didn't even realize that checks out though. That checks out.
Oh, that's supposed to be like a secret. Yeah, you have
I guess
70.
I guess I could like count, like look at the, but I
I know you have an assignment like every class.
basically
At least one.
at least
Yeah.
Are these are your age or these are your samples?
These are my tablets.
You can just pack them up and then you can take them with you and just look at them over.
Yeah,
Oh, nice.
look at them like maybe. Wait, this is with
Your room temperature.
Yeah, just,
Yeah.
like don't leave it at this, like don't go out.
yeah, don't bake it in a UV light.
Like just put it in your pocket and then you
Okay.
just,
You could, yeah, maybe if you like put it somewhere warm, like 37, you can speed up the reaction.
It definitely works at higher temperatures. So we just don't require it.
Yeah.
I was like at room temperature, we should like slightly see something already after 1 hour if you want us we can. But definitely after 3, you should be able to see something.
you guys hand fill these for every workshop that you do.
Just this class, actually.
We had a
But yeah,
Question.
also.
question, yeah.
Yes.
He said just little clarification. Is this about color? Is this like a color change, like a lamp test? And what are you currently testing or working on?
Did I type it or
I think you can, like,
are they ready for the answer? Right.
and then we had another question. Okay, yeah, we'll just do this one. So is it like a lamp test? I guess what's the difference between this and a lamp test?
I'm not quite sure what a lamp test is, but it's a sort of simple idea where essentially the light on is just the regular biological state, and then the CRISPR is essentially turning off the light for the color in this situation. So it's sort of like a lamp test. And then the second part of the question was, I forgot.
What are we currently testing?
So we're testing the production of GFP and how CRISPR can
Not like this. Like,
essentially turn off production of GFP or honestly any gene of interest. But we're using GFP because there's a visual way of seeing that the gene is essentially being deactivated in a situation.
Because like if like you go up like volume is split up, right? So your reactions split up.
If they don't like the answer, they can.
No, I think that was good. So Kennedy, thank you still for being online.
So like
We had this
Hold it.
Okay.
sheet with the protocol. Basically, we have these 4 reaction
And then
tubes.
like maybe like
Might just like go up with
2 of them are, or 3 of them are controls, and 1 of them is our actual test. And we hope to see that in our guide, we introduced a GFP protein, so in theory it would fluoresce, but we're actually blocking it with this dCas9 and guide RNA. And so the results would be a no color or a clear color for the guide reactant, while these 3 would, the first and the third.
The first one is negative, like should have no color.
Yeah.
The second one has the GFP, so it should be a color. The third one has the GFP and only the dCas, not the guide RNA, so it should also have a color. And the guide RNA has the plasmid, like the GFP, the dCas, and the guide RNA, so it should not have a color.
Okay. So 1 and 4 should be clear, 2 and 3 should actually be green.
And here you can see it again here.
Oh yeah.
on
Yeah.
the slide. Yeah.
Okay.
It's sort of cheating because we asked the students what color they hypothesize the tubes to be, so like that.
So a bit more expedited.
This is such it. Wait, so you said you don't hand fill the tubes normally?
We do.
Oh, you do?
No, we usually don't. We go to classrooms and students actually sort of do it themselves. And one thing they're practicing is just like basic pipetting.
What about your samples?
I think part of what makes this experiment
you know,
interesting
you
is like the
showed us
hands-on component of it, right? If we sort of just put everything in those type of fix it, it's kind of
Flipping one you
Fair
don't
enough,
need
fair
to.
enough.
You're flipping one that you don't need to do.
I'm filming one that I don't need to.
Yes, because that's your process to advance with place study. Obviously, you don't need to.
Oh, okay.
You edited GP, right?
Yeah, yeah.
Okay.
Are you sure about that?
Very nice.
Wait, how do you pipette again?
Don't ask me.
Check it with the wallet before it's used. So
When I click down, it doesn't.
Huh?
When I click down, it doesn't.
Well, like that's like the small ones are pretty hard to get them. Yeah. It's mainly about the big drops, and like you see, like you do have like a good volume down there, so like it's.
Yeah.
anyone want CRISPR fit stickers? I have like a couple of them.
Yeah.
If you want, you can have one. Okay.
Thanks.
How big is the team that
Um,
works on this industry?
Right now it's about 8 people, but we're recruiting. So if anyone is interested, we're interested. So yeah.
Oh, it makes sense. I don't see any green yet.
Oh no, it takes like around 3 hours.
Okay, so then
3 to 24 hours.
I guess I don't know how it's working, but well, no issues. Yeah.
Yeah, just like
So just by, so like in this lighting, we should be able to see green.
Yeah. It might even glow in the dark. I haven't tried those yet. Though.
That's cool.
I also have like one ready like in the left. Should maybe brought those.
Okay.
Yeah, we could have brought one as a reference.
I'm gonna head out.
Well, thank you so much for.
Oh yeah, thank you so much for having us.
Yeah,
And
thank you guys. Bye on Zoom. We'll see you on Monday.